Assalamualaikum and good day, everyone. Have you ever wondered how scientists can analyze thousands of genes in one go? Well, today we are going to dive into the incredible world of DNA chips and unravel the mysteries behind this amazing technology. Let's go!
DNA microarray analysis involves breaking open a cell, isolating its genetic content, identifying all genes that are turned on in that particular cell, and generating a list of those genes. A DNA microarray, also commonly known as DNA chip or biochip, is a collection of microscopic DNA spots attached to a solid surface. Scientists use DNA microarray to measure the expression level of large number of genes simultaneously or to genotype multiple regions of a genome. DNA microarray analysis is one of the fastest growing new technologies in the field of genetic research. So let's take a look for significance of DNA microarray. The first one is a high throughput analysis. DNA microarray has revolutionized genomic research, allowing for large-scale studies that were previously impossible. The second one is cancer genomic. DNA microarrays identifying specific genes of cancer and helps in early diagnosis as well as in monitoring disease progression and response to treatment. The third one is targeted therapies. DNA microarrays identifying genetic mutations and variations associated with specific disease. And the last one is gene regulation by helping in understanding how genes interact and regulate each other.
Now we move to the problems and optimal approach. One of the greatest challenges in biology is understanding the molecular mechanisms behind normal and dysfunctional biological processes. DNA microarrays enable us to simultaneously analyze the structure and activity of thousands of genes and proteins. This high throughput approach provides invaluable insights into complex biological systems. In clinical settings, diagnosing complex diseases with multiple genetic factors can be daunting. DNA microarrays offer a solution by identifying gene expression pattern associated with diseases. This capability facilitates early detection and enables personalized treatment strategies. Traditionally, studying gene expressions in organisms required laborious gene-by-gene methods. DNA microarrays now allow researchers to monitor the expression of all genes in an organism simultaneously. This holistic approach accelerates our understanding of cellular physiology and disease mechanism. Next, efficient screening for genetic variations and biomarkers in disease like cancer is crucial for targeted therapies. DNA microarrays excel in identifying differentially expressed gene and biomarkers, paving the way for precise treatments and improved patient outcomes. Next, environmental stresses can have profound effects on an organism's genomes, but their analysis is complex. DNA microarrays enable researchers to assess genome-wide expression changes in response to environmental factors. This provides insights into how organisms adapt and respond to their surrounding. Last but not least, the detecting pathogen infections early is critical for effective treatment. DNA microarrays detect changes in gene expression indicative of pathogen presence, facilitating the development of rapid diagnostic tools and targeted treatment strategies.
DNA chips offer several advantages in forensic science. DNA chips and their associated scanning equipment offer the advantage of being relatively compact and portable, making them suitable for use in various locations, including on-site forensic labs. They can also provide precise identification by detecting specific genetic variations despite working with very small amounts of DNA. In terms of speed, DNA chips can generate results more quickly than traditional methods, with the rapid screening of suspects or identifications of victims. This rapid generation of results speed up investigation and legal processes. Next, DNA chips can analyze thousands of genetic markers simultaneously, providing comprehensive genetic profiles. This capability allows for the rapid processing of multiple samples, which is particularly useful in complex cases involving many pieces of evidence. Automation in DNA chip analysis reduces the chance of human error and increases consistency. The use of standardized protocol across DNA chips enhances the reliability and reproducibility of results. While DNA chips offer significant advantages in forensic science, they also come with certain challenges and limitations. Setting up the necessary infrastructure and purchasing DNA chips and scanning equipment can be costly, with additional expenses for maintenance and ongoing chip purchases. Other than that, the accuracy of the results obtained from DNA chips may be affected, as DNA from crime scene is often degraded or contaminated. Forensic samples may contain DNA from multiple individuals, which will complicate the analysis and interpretation. Moreover, operating DNA chip technology requires specialized knowledge and training, as well as bioinformatics expertise, due to the complexity of analyzing and interpreting large volumes of data. These experts may not be readily available in all forensic labs, posing a challenge in accessing specialized professionals for DNA chip technology. Due to their high sensitivity, DNA chips are susceptible to contamination from handling or the environment, which can potentially lead to false results if not carefully managed. Lastly, the use of DNA chip technology must be validated and accepted by the legal system, which can vary by jurisdiction. The handling and storage of genetic information raises privacy concerns that necessitate strict ethical guidelines for addressing these issues.
The technology has potential uses in forensic science, particularly in the areas of forensic genetics, forensic pathology, and forensic intelligence. With DNA microarray, entire microbial genome can be easily represented in a single array, and it is feasible to perform genome-wide analysis. Despite its potential in forensic science, microarray technique is also used in medicine development by providing microarray data of a patient, which could be used for identifying diseases such as cancer, influenza, and gastrointestinal viruses. DNA microarray has been used for studying gene expression analysis in neurological disorders. DNA microarray experiments are carried out to find genes which are differentially expressed between two or more samples of cells. DNA microarrays can be used to analyze DNA evidence left on objects or in environments such as crime scenes. By using microarrays, forensic scientists can identify and quantify specific genetic markers that can help in identifying individuals or linking suspects to a crime scene. This can be particularly useful in cases where the DNA evidence is degraded or only small amounts are available. Additionally, DNA microarrays can be used in forensic pathology to analyze gene expression patterns in tissues obtained during autopsies. This can help in determining the cause of death, especially in cases where the cause is not immediately apparent. By examining the expression levels of certain genes, pathologists can infer the physiological state of the disease and potentially identify markers of disease or exposure to toxins. Other than that, this technology allows for the analysis of large numbers of genetic markers known as single nucleotide polymorphisms (SNPs), which can provide valuable information for criminal investigation and forensic casework. Forensic genetic genealogy, which DNA microarrays are used to generate high-density SNP profiles that can be compared to genetic genealogy websites or databases. This approach has been successful in identifying suspects in cold cases such as the Golden State Killer and the 11M Magic Bomb attack investigation by finding distant relatives of the perpetrators through genetic matches. Next, kinship analysis, which DNA microarrays can determine the degree of relatedness between individuals, which is crucial in identifying missing persons, establishing paternity, or linking suspects to a crime scene through familial relationship. Furthermore, direct analysis from casework like samples which is the study you provided demonstrates that DNA microarrays can generate reliable SNP profiles directly from forensic sample without the need for DNA purification, potentially simplifying the analysis process and reducing the costs.
I will now compare traditional forensic DNA profiling with DNA microarray profiling. Traditional methods rely on short tandem repeats or STR analysis, requiring DNA purification and PCR-based capillary electrophoresis. It is highly effective for individual identification, paternity testing, and criminal investigations. In contrast, DNA microarray profiling uses high-density single nucleotide polymorphism, or SMP profiling. It simplifies sample preparation with direct protocols and leverages high-throughput microarray platforms. This method enhances forensic intelligence, providing insights into biogeographic ancestry, appearance prediction, relatedness, and sex determination. Next, we look at the strength for both of them. Traditional forensic DNA profiling is highly reliable and widely accepted in courts, thanks to established protocols and database. It is particularly suitable for small degraded samples due to the sensitivity of PCR. On the other hand, DNA microarray profiling offers higher resolution by analyzing a large number of SMP. It can provide additional information on ancestry and physical traits, making it useful for forensic intelligence and linking suspects through familiar connections in commercial database. Next, let us look at the disadvantage of both profiling methods. Traditional forensic DNA profiling is limited by the number of STR markers, typically 13 to 20 loci, and provides less information on biogeographical ancestry and physical traits. It's also labor-intensive, involving multiple steps. On the other hand, DNA microarray profiling requires higher DNA input, posing challenge with degraded samples. It is more complex and costlier than STR profiling, and there are ethical and privacy concerns with using commercial genetic database.
To further advance DNA chip technology, several recommendations can be considered. First, enhance sensitivity and specificity. Develop improved prop design and surface chemistry to enhance hybridization efficiency and reduce cross-hybridization, thus increasing the accuracy of detection. Next, integrate more advanced fabrication techniques to produce higher density arrays, allowing for the analysis of more genes or genetic variants simultaneously, and implement faster hybridization and detection methods to reduce processing time. Moreover, establish standardized protocols for manufacturing, processing, and analyzing DNA chips to ensure reproducibility and readability of results across different laboratories. We also can develop robust quality control measures to monitor and maintain the consistency of DNA chip performance. Integration with other technologies also can be done. Combine DNA chips with next generation sequencing, NGS, and other genomic technologies to enhance the breadth and depth of genetic analysis and explore the integration with lab on a chip device for more comprehensive point of care diagnostics. Lastly, design DNA chips tailored to specific populations or diseases to improve the relevance and applicability of results in personalized medicine. Also, focus on the development of chips that can analyze multiple biomarkers simultaneously for comprehensive health assessment. In conclusion, DNA chips, also known as DNA microarrays, have revolutionized the field of genomics by allowing the simultaneous analysis of thousands of genes. This technology involved the hybridization of DNA samples to a solid surface embedded with non-DNA sequences, facilitating the detection and quantification of gene expression level and genetic variation. DNA microarrays have been pivotal in various applications, including gene expression profiling, disease diagnosis, and the identification of genetic mutations. The use of DNA chips in gene expression profiling has provided insight into cellular response, disease mechanism, and the effect of treatment at a molecular level. Moreover, advancements in microarray technology have improved sensitivity, specificity, and throughput, making it an indispensable tool in both research and clinical diagnosis.